Hello, hello. Hello everyone. Today I am. It is week 9, and mostly last class I have taught about the different like rearrangement of both uh, B cell receptors as well as T cell receptors, how they join together to form the complete receptor, like how diversity is created to uh, recognize different kinds of pathogens. And then in the last, I have also talked a bit about uh, different kinds of infections which can happen to us. So today I will continue about uh, this only, this infection. So today, now Sir is teaching in week 9 about the immune responses to mainly viruses. So before going on to virus, I will a bit talk about the different kinds of pathogens and the different kinds of defenses which we are having. And there has been doubts, only one doubt this week, which uh, someone has posted in the portal. So I will discuss that doubt in the last few minutes of this session. If someone so if people are joining, I will also ask what are the doubts they have. So let's begin. So basically, now I'll be teaching about the infectious diseases and vaccines. I hope people know what is infection before going on to infectious diseases. So basically, infection means like it is a person is said to be infected when that person has been invaded, like the host system has been invaded and there by some pathogen or some germs, like it can be anything like bacteria, virus, fungi, yeast, anything. And if a particular person is has become like, it is known as the invasion of the different kinds of tissues, their multiplication, their growth, their the reaction of the host to the infectious agent, and uh, the toxins that are produced by the host system. That infection is the whole. This process is known as infection. And diseases which are infectious, like diseases which are caused by this process, are known as infectious diseases. And vaccines, about you all know what are vaccines. Like I will come to this part later. First, I will speak about the infectious diseases. So I hope everyone remembers about the cowpox inoculation, which we have studied in the beginning of our course, uh, regarding like when there was a smallpox uh, inoculation from cow, like like cowpox inoculation helped the people from surviving through smallpox. And this is a sculpture you can see, which was in the period 1644 to 1911, this king period. And this is like how there was inoculation, like this is a woodcut showing sites for cowpox inoculation from an 1888 edition of Definitive Treatise on Pox Diseases by Zhu Chungxia. So it was one of the earliest story where smallpox was prevented by cowpox inoculation, and we all know how. Edward Jenner has eradicated, like not eradicated, like he has uh, inoculated the boy similarly with who was having uh, rabies, who like rabies virus. So uh, like that way from the beginning, it has been this treatment of these different kinds of diseases, where in practice now we are having modern medicines, modern vaccines. So now we are much more controlled, like all these diseases are much more controlled compared to the earlier times. So here is a list of the top 10 causes of death all worldwide among children under 5. So here you can see what are the top 10 causes of death. First, it is preterm birth complication. Second, it is lower respiratory infection. Third, it is birth asphyxia and birth trauma. Fourth, it is diarrheal disease. Fifth, it is congenital anomalies. I hope everyone knows preterm birth complication means before uh, bo- like before birth there can be different types of complication with the mother also, with the position of the baby also, or other complications. Lower respiratory infection mainly by the viruses which we see. Birth asphyxia and birth trauma means by chance if a person during before birth cannot breathe or there's some kind of asphyxia occurs, then it might lead to death. Then diarrheal diseases is a common thing. Congenital anomalies means uh, it can be like it is present only from the hereditary things. So you can see all the other blue color means either it is communicable maternal, perinatal and nutritional conditions. These are affected, and only this congenital anomalies is a non-communicable disease. So other is neonatal sepsis and infection, like the infection if it spreads throughout the body it leads to sepsis. Then it can be malaria, it can be malnutrition, it can be measles, meningitis. So there are different kinds of uh, diseases. These are 10 topmost diseases which is causing death among children under 5. So so here we since we are talking about the infectious disease, an infectious disease always involves any kind of pathogen, and so we will first talk about the possible ways by which pathogen can enter our body. It has already been taught, I think, in anatomical barriers. I have taught, Sir has taught. So here you can see, like we know for the foremost thing which allows a pathogen to enter into our body is the skin, and there can be any mucus lined surfaces which act as a buffer between host and infectious agents. There are different kinds of barriers through which the pathogen should enter into our system, but these barriers protect us from thousands and thousands of pathogens every day. So sometimes apart from this pathogen itself, there are certain other living organisms which help these pathogens to enter into our system. So this is a third party, and this is known as a vector which helps to carry the infection from one organism to another. I know that everyone knows what a vector is, and you guys can easily tell about the about an example which we see every day in our like around. So so the vectors are or it can be named as intermediate hosts. They help to transmit this infectious or to help the infectious agents to get into the host system by circumventing the host barriers. So they transmit this infection from one organism to another. So they can be known as intermediate host. They can transmit a pathogen between one infected human and another, or from an infected animal to a human. I need not say the example. Here you can see the example. Infectious disease vectors are often blood-sucking arthropods, example ticks, fleas, flies, or mosquito, which breach natural barriers like the skin with their bite and can introduce the pathogens they carry into a susceptible host. The most common things we can see every day in our naked eye is the mosquitoes, which can actually harbor lot of pathogen inside it, and it can cause different types of disease. Though different mosquitoes cause different types of disease, not the same mosquito will cause which will cause malaria will cause dengue, but still. So these diseases infections are known as vector-borne infections, and they approximately account 1 out of every 6 instances of human infectious disease and are referred to as restricted to areas in which the intermediate host is found. Example include malaria and Zika fever. So all these are caused by mostly mosquitoes, and the first responders of the innate immune system are an additional barrier to infection, sometimes clearing pathogen without the need for an adaptive response. So we know innate immune response is the first barrier or the first line of defense system, while adaptive response is much slower compared to the innate one. So sometimes it happens that innate immune system is enough to actually restrict the pathogen inside our system, and it can clear the pathogen without even activating the adaptive immune response. So these are the like different kinds of vectors you can see. Number one is flaviviruses. Example include yellow fever, Japanese encephalitis, West Nile virus, dengue virus, Zika virus, chikungunya virus, Onion virus. The most common in our India is obviously dengue among this. So the vector of all these viruses is are the mosquitoes, and the genus or species are Aedes or Culex. So Aedes aegypti is the carrier for dengue, and Culex also can cause other like Zika. So in case of bacteria, there can be Yersinia pestis, plague, Rickettsia prowazekii, typhus, Rickettsia example Rocky Mountain spotted fever, Borrelia, Lyme disease. The vector in these cases are flea, lice, ticks, etc. And the genus or species are like the Xenosylla, Pediculus humanis, Dermacentrixoides. These are the different kinds of genus and species of this all these insects. Like flea, this plague is caused, and I don't know whether you all know there was a very much outbreak of plague. Like it was a huge population actually suffered. Bubonic plague it is named as, and this is caused by the genus Xenosylla of fleas. And lice, you all know what is lice. Ticks. Then apart from that, there is protozoa. There are plasmodium, which causes the malaria, and the carriers are again the mosquitoes. Here, the genus is different from the chikungunya or Zika or dengue virus, and it is Anopheles one. Then there is trypanosoma. There is Trypanosoma cruzi, which is the Chagas disease, and they are caused by kissing bugs. Genus includes, they are not caused by, they are carried by kissing bugs. They are Rhodnia, striatoma, these are the genus. Then there is Trypanosoma brucei, which causes the sleeping sickness. There are tsetse flies, which are carrying this protozoa. Then the genus includes Glossina morsitans. Then there is, apart from that, there are leishmania or leash, which is called leishmaniasis. It is carried by S flies, and the genus includes Phlebotomus lutzemium. And also there are filarial worms. So these are the different kinds of pathogens which are carried by different kinds of vectors and cause different kinds of diseases. So there is a link between the location and the immune effector mechanism. So all these diseases which are caused by different kinds of pathogens, whenever they enter into our body, it depends where it is entering and which immune responses they are activating. So not all the pathogens which enter will induce a similar type of immune effector mechanism, and not all the pathogens which enter into our body will enter through the same location. So there is a link between the location as the immune effector mechanism. For example, infections can occur on body surfaces, for example in skin or mucosa, or they can penetrate the epithelial layers, breaching skin, urogenital, or gut linings. Through these openings, they can easily enter into our system. So these are the barriers which protect us from the outside pathogens or the outside world of pathogen. Once these pathogens cross these barriers, they can they can stay at that particular place from where they have entered, or they can spread from the site of entry. So uh, there are certain infectious agents which are found in the interstitial fluid, which are between which bathes our tissues, means the tissue fluid, and from there they should then be carried to draining lymph nodes. So there are many infections that actually can enter our bloodstream, which is very rare. But if it enters, it can lead to a condition known as sepsis, which is life-threatening. So when this happens, when the sepsis occurs, the infection can spread quickly throughout our body, and our immune system, what it can do, it can do more harm uh, than harming the pathogen. It can do more harm to the host itself, and it can lead to septic shock. So I have discussed last week what is septic shock and what is sepsis. Like, this condition is extremely rare, not every time it happens, but if it happens, it is very much it is not a good sign. It is life threatening, and there will be too much abrupt changes in your parameters like heartbeat, blood pressure, etc. And it it leads to their death if not corrected at the correct time. So there are some pathogens that can enter into interstitial fluid. There are some pathogens that can enter into a bloodstream. And there are other pathogens which spend all or part of their lives inside the host cells, occupying either the vesicular or cytosolic and nuclear spaces. So there are pathogens which like to remain dormant and they want to live inside the host cell. They don't want to do anything, just live inside the host cell, but they can become active at a later point. They can go and occupy anything like cytosolic, they will form inside the vesicles, they can form, like they can just sit there, or inside in the nuclear spaces they can remain. Importantly, the entry site and ultimate location of an infectious agent in or on the body will determine which immune tools are available and best suited for pathogen detection and elimination. So, where the pathogen is entering our body is important, and where it is going, that is, the ultimate location is important, and these location will decide which immune tools or which immune response will be generated and is best suited for pathogen detection and elimination. So, so first, most infectious organisms enter through the mucosal surface, as I told, like the respiratory. When we are breathing, the anything from the air can enter into our respiratory system, or through gastrointestinal system. Like whenever we are eating something, it can enter into our gastrointestinal tract. Like we eat so many things, like foreign things in our body, like we are taking, but so it should be like so many pathogens can enter at that time, and or through urogenital tracts whenever we are going to any bathroom or something. uh, it can There are millions and millions of bacteria which are there, or other uh, like pathogens which can enter. So mostly these organisms enter through these points. Here you can see through inhalation, respiratory tract, through ingestion, gastrointestinal tract, or through contact also, conjunctiva, through your like fluid, that eye fluid, it can also enter. Then the urogenital tract is there. Then once these pathogens have crossed the barrier and they have uh, s- like they can also enter through some reaching the skin barriers through a wound or via an insect bite. So whenever there is a wound, I hope many people have heard that doctors tell that you try to cover the wound. This is why we put band-aid, or why we put any kind of like protection around the wound, because any kind of breach is there, any kind of pathogen can enter into your body. So these pathogens wait for any kind of breach in skin barriers so they can enter. Or through an insect bite also these pathogens can enter. So once they have crossed this barrier, they exist in the sterile microenvironment of the body, where they can remain local or spread through blood and lymphatic circulation. So whenever they are breaching, they have crossed this barrier, they have breached the barrier, they can exist in the sterile microenvironment of the body. So there they can either remain in the same place, or they can spread through the blood and the lymphatic circulation. So there are different types of examples of all the four types of pathogens which are there. The four types of pathogens include bacteria, fungi, protists, and virus. Bacteria, it can cause ear infection, pneumonia, food poisoning. Fungi can cause ringworm, athlete's foot. Protists can cause malaria, and virus can cause like common cold or flu or AIDS. So these are the four different types of pathogens which are capable of entering and interfering with our immune system. So after Like examples of all four types of pathogen can be found in the extracellular fluid, while intracellular parasites and bacteria may be involved or they can enter the host cells and remain vesicular. So these all four types of pathogens which I told, bacteria, virus, fungi, and protists can be found in the extracellular fluid, but there are intercellular parasites which can enter uh, enter or they can be engulfed, like parasites and bacteria. They can be engulfed or they can enter the host cells and remain vesicular. In vesicles they can stay. Here also, as I told, you can see when the wound or arthropod bite is there, the pathogens can enter. Then these are the different. M means mucosal, E means extracellular, V means vesicles, intracellular, C means cytosol. They are also intracellular. So these are the different. After breaching of the epithelial barriers, you can see the virus can be in the cytosol, or they can be in the extracellular fluid also. The bacteria can remain inside the vesicles. The helminths also in the vesicles they can live, and the parasites also can reside in the vesicles. So different kinds of like for example, here you can see virus fuse with the plasma membrane and enter the cytosol, from where they may insert their genetic material into host chromosomes in the nucleus. Here you can see it can. Some viruses are there, especially DNA viruses, also some RNA viruses like HIV. They can actually enter into the nucleus and they deposit or they insert their genetic material which combines with our own DNA, and it can live like this forever, kind of. But sometimes it becomes active also. So each of these locations allow for access to different detection molecules and may necessitate different effectors for elimination. So these four different four different sites like extracellular, vesicular, mucosal as well as cytosolic. These four different sites require for different kinds of effectors. Like these sites will have different effectors which will be eliminated. So, for example, in case of mucosal surface, through mucosal surface, anything can enter. Example include virus, bacteria, helminths, and fungi, and they can be innate lymphoid cells, malt B and T cells, soluble IgA, mast cells with IgE, antimicrobial compounds. So the mostly these Th2 responses come into place wherever there is some entry of the pathogens to the mucosal surface. Then the extracellular surface, there can be bacteria, helminths, and fungi which can enter into the extracellular fluid. Then the different cells will be Th2 and Th17, and Th2 or Th17 cells, there can be response from the B cells, there can be response from the Ig, IgM, and IgG. There can be phagocytic cells or antimicrobial compound which can be secreted. Then there are vesicular uh, or the vesicles through which like the Th1 response, these vesicles, mostly these bacteria will and parasites will come and stay in the vesicles, and mostly the effector cells are the Th1 cells, the cytokines which are released, there are macrophages, there are NK cells. Apart from that, there are pathogens which enter into the cytosol, mostly viruses and some bacteria, and the type of immune response is mediated by Th1 cells and cytotoxic T lymphocyte cells. So here you can see CTLs, Th1 cells, cytokines, and NK cells and ADC, ADCC cells. Uh, so ADCC is like antibody dependent cell cytotoxicity, means by which antibody like go and bind to the antigen and then they activate the natural killer cell and kill the infected cell, kind of. So these you can see different kinds of pathogens enter different through like enter through different sites, and then it can uh, kill, or it can synthesize different kinds of molecules, and it can attract different kinds of molecules which mediate the immune response. And it all depends on the location of the entry, the first the entry from where a pathogen is entering, then the location where the pathogen is still there, and the kind of uh, response which the immune system is generating. So these are the four different pathogens. So there are mucosal or barrier infections are typically controlled by Th2 type responses. The majority of but not all infectious agents enter through mucosal routes. Example: mouth, nose, eyes, or urogenital tract. When a would-be pathogen enters the digestive system, so mostly infectious agents enter through our eyes, nose, mouth, or urogenital tract. uh, but sometimes it enters through our digestive system. Uh, but when it reaches like suppose it is entering through our mouth, it should reach the digestive system. So digestive system, we know that there is acidic. Our stomach is acidic, and there is acid inside our stomach. So it should survive that stomach acid first. Then it should make through way through the intestinal tract. But if the stomach acid is capable of killing the pathogen, so it won't be able to reach the intestinal tract. So every step there is a layer. First, it has to make a breach into our like entry route, the mucosal route, or the other routes. Then it has to survive the stomach acid. Then only it can get into the intestinal tract. Then only it can go into different kinds of locations. So at every step there are different kinds of layers through which that pathogen has to pass. So that is why it is not very easy for a pathogen to enter into our body, because our body is also composed of different kinds of layers which is actually protecting it, like us, from the different kinds of pathogens. Then immune effectors are there, which include soluble antimicrobial proteins which are secreted by epithelial cells, innate lymphoid cells, and different kinds of and a dispersed set of lymph node-like structure which is known as mucosal associated lymphoid tissue. Malt and gold gut-associated lymphoid tissue and mucosal-associated lymphoid tissue, which we have already talked previously. These are the different lymphoid cells which are present in different locations. For example, in case of mucosal surface, if the lymphoid tissue present is known as mucosal-associated lymphoid tissue. So these tissues secrete a large number of antimicrobial proteins, and they will protect us from the different kinds of pathogens. Then pathogens that reside in or near the surface of the body are specific controlled by Th2 type of responses. For example, they include the ILC2s, Th2 specific cytokines. And then there are IgE, which is capable of recognizing surface epitopes of the pathogen. So mostly which reside near the surface of our body are controlled by the stage 2 responses. Then immunity against many of the most common metazoa parasites, for example helminth or worm, correlate with high antigen specific ratio. For example, there should be a high ratio of IgE to IgG. So uh, whenever there is any parasite infection like helminth or worms, so the ratio of this IgE to IgG will be more. So from there also you can detect what kind of infection you are having by just doing a test. Then the cells, for example, basophil eosinophil mast cells will increase the expression of this IgE. IgE is what? Immunoglobulin E or the antibody. So they will increase the IgE receptor and they will mediate a Th2 type of response. Another player important which is in the health of various surfaces is dimeric IgA or DIGA. This particular isotype is found most abundantly at mucosal surfaces after being carried across epithelial barriers via transcytosis or by the poly-Ig receptor. So another important player is IgA or immunoglobulin A. Mostly it is found in our saliva or the mucosal surfaces, and they can cross the epithelial cells via process transcytosis. I have also spoken about what is transcytosis. So transcytosis is like a process in which there is a specialized transport to which a particular molecule which is coming from outside is endocytosed. Then they are sent from one place of the cytoplasm to another area through membrane bound vesicles, and they are secreted at a different plasma membrane surface. So this is a process known as transcytosis, and this is a process by which this IgA can travel from one part to another. Then this IgA can bind to the infectious agents and block the attachment of these agents to epithelial cells, thus neutralizing them and help in the elimination of them passively without any inflammation. So this is a good thing that without inflammation, the IgA is able to control the infection and is clearing of the pathogen. Because as I already told, excessive inflammation can sometimes cause many diseases or like autoimmune diseases, and it is not good for health if there is excessive uncontrolled inflammation. So without any inflammation, this IgA can help in removing the pathogen from our body. So extracellular pathogens, for example, what are the extracellular pathogens we learned? Extracellular pathogen includes bacteria, helminths, fungi, or it can be viruses also, which can remain in the extracellular fluid. So this extracellular pathogen can be recognized and can be attacked using extracellular tools. So they can remain local or spread via the either via the circulatory or the lymphatic system. So they will mainly move through the fluids and they can spread to the other part of the body through the circulatory and lymphatic system. Extracellular infections within the body are controlled by different like combination of both innate as well as adaptive effector mechanism present in extracellular fluid, for example, phagocytosis and APC activation. For example, there is binding of these PRRs or the pathogen recognition receptors, and there is production of cytokines. Can be complement activation, and there can be antibody binding. So it requires both adaptive as well as innate immune system to control the extracellular infection which are present in the extracellular fluid. So all classes of pathogens that breach epithelial layers will be found in the extracellular spaces, even for example, even viruses for at least some time of their lifetime. Therefore, these extracellular immune effectors can do a play in recognition, neutralization, eradication whenever there is presence of this pathogen in these spaces. So both adaptive and immune response is needed to clear off the any kind of extracellular infection, and they can mm, like for example, virus can sometimes stay in the extracellular space before going into the cytosol or any nucleus. So these extracellular pathogens are controlled by different kinds of immune effectors and which play a role in recognition, neutralization, and eradication whenever there is a chance to do so. Uh, these remains this when about the extracellular pathogens. And there are intracellular pathogens, which are the intracellular pathogens. Intracellular pathogens include all like bacteria, parasites, viruses, etc. Should these intracellular parasites must be recognized, and there are different kinds of mechanisms through which these are recognized. For example, infectious organisms that reside inside the host cells are very much difficult for the immune response to find and destroy. So whatever uh, intracellular thing which has entered into our body and is residing like without creating much problem in our body, it is very difficult for the immune system to understand that a pathogen has entered into our body if it cannot recognize the pathogen properly. So they are difficult to find, and it is difficult for the immune system to destroy these pathogens. So examples include intracellular bacteria, intracellular parasites, and both of them are smaller than the extracellular counterparts, and all viruses which are obligate are intracellular pathogens. So obligate means uh, uh, what to say, which is obligate virus obligate pathogens means which cannot survive without the help of a host. Like it will fail to reproduce or it will fail to even survive if there is not a proper host. So the many viruses and other parasites also, there can be parasites which are obligate, which are unable to live extracellularly, and they can only multiply or they can produce more and more virus of their progeny only when they are intracellular in nature. So uh, most bacteria and parasites that are intercellular spend at least some time with intercellular vesicles, where they might trigger different kinds of PRRs or pathogen recognition receptors such as TLRs or toll-like receptors. So there are certain bacteria and parasites which spend sometimes within these intercellular vesicles, and they activate the PRRs, which are known as the toll-like receptor or TLR. So, if we want to eradicate these infectious organisms that occupy these endosomal spaces inside host cells, we require a strong stage 1 pathway response. While you can see that extracellular pathogens are mostly controlled by Th2 or Th17 response, while intracellular ones are controlled by this Th1 response. So two types of response depending on the site of the entry or the site of the localization of the pathogens. So there is a phenomenon which is known as a delayed type hypersensitivity, which occurs. It is a case uh, where cytokine is secreted by Th1 cells, and it delayed type, why it is called, because the reaction occurs after almost 24 to 72 hours after being like exposed to a particular antigen, and so this is known as delayed type hypersensitivity. Like the response is much slower, so it's delayed, and it relies on the secretion of the cytokines by Th1 cells. Example includes IF and gamma to activate different macrophages that can digest these pathogens, which are vesicle bound and can overcome common pathogen induced immunization of phagosome lysosome fusion. So Sorry for the interruption. There was an emergency, kind of. So, um, uh, uh, so this is a phenomenon known as delayed type of hypersensitivity, in which there is a delay response in response to a pathogen, and there is cytokine secretion by Th1 cells. Example include IF and gamma to activate macrophages that can digest these vesicle-bound agents, and that can overcome different common pathogen-induced immunization of phagosome lysosome fusion. So, intracellular infections are very much difficult for the immune system to detect and eradicate, and they can be divided into membrane bound and cytosolic with differing key immune response mediators. So, intracellular, obviously, as I told, like the parasites and bacteria, they will be very small, and then their extracellular counterparts. So, they are very much difficult to identify by the immune system, and eradicate them. So there can be different kinds, for example, membrane bound cytosolic, and they have different immune response mediators. Intracellular vesicular infections, thus, are mostly effectively eradicated by macrophages, activated by cytokines secreted by Th1 cells, while cytosolic infection requires host cell lysis by CTLs or cytotoxic T lymphocytes, uh, cytotoxic Th1 cells or NK cells. So, intracellular and extracellular pathogens are removed differently from our system, and there are different cells which are involved in these two types of different pathogens. So, for example, there is a example, as you all know, TB or tuberculosis. It is a disease which is caused by the pathogen or a bacteria, which is Mycobacterium tuberculosis. Mostly it is seen in 90% of infected individuals, like it causes pulmonary tuberculosis. So tuberculosis mostly is seen in immunocompromised people now, especially in people having AIDS or HIV. They are more susceptible to this tuberculosis infection, and there is a chance of pulmonary tuberculosis. And in case of this infection, we can see there is an activation of CD4 T cells, which are activated within 2 to 6 weeks of infection, and they can secrete cytokines that induce infiltration of large number of activated macrophages. So then what happens? These macrophages are getting activated, and the cell wall of the organism inside the granuloma, which is called a tubercle of a cluster of small lymphocytes surrounding the infected macrophages. So this a granuloma kind of structure is formed, which is made up of small lymphocytes, cluster of small lymphocytes which surrounds this infected macrophages. The There is a localization of the lysosomal enzymes inside the granuloma that can cause excessive tissue necrosis. So this granuloma is formed in this kind of tuberculosis infection, and there is massive activation of macrophages that occur within these tubercules and result in the concentrated release of the lytic enzymes. These enzymes will destroy the nearby healthy cells, resulting in circular regions of necrotic tissue, which eventually form a lesion with a cheese-like consistency. So the enzymes which are secreted will destroy also the nearby cells, resulting in circular region of necrotic tissue. They will form a lesion and it will be destroyed. After these lesions are healing, they become calcified and then can be readily visible on X-ray as a defined shadow. So, by that you can see to what extent your lungs has been damaged, and Much of the tissue damage seen with M. tuberculosis is thus actually due to pathology associated with the cell-mediated immune response. So here you can see, compared to the damage caused by the uh, pathogen, it is caused by the activated macrophages, and that much like this pathology you can easily identify in the x-ray, and you can understand like that much destruction has occurred that you can easily identify it via x-ray. So here it is a tubercle or a granuloma formed in pulmonary tuberculosis. Here you can see the bacilli or the bacteria is present, is in the center. Then this is macrophage with the bacilli. These macrophages are forming a layer around it. This then these are the activated macrophages here. Then this is the Th1 cell in between. Then you can understand this is a structure which is formed, and there is too much cytokine release, and they will this during this pulmonary tuberculosis Th1 cells will recruit and activate the macrophages which surround the dead and dying infected cells. Here the dead and dying infected cells that harbor the bacilli, like these bacillus has entered into the macrophages and other macrophages coming and surrounding it, thus uh, preventing the bacteria to spread further into different uh, zones, and these is secreting many lytic enzymes which is destroying the bacteria as well as the cell, and you can understand the lung gets damaged and you can see the it as a x-ray. So this Th1 response is mainly helping in fighting the intracellular bacterial infection. So before going to other kind of infection, I will first start with the viral infection. And before going into details how viruses are being cleared by the immune system, I hope like people already know different kinds of viruses, and there are different viral infection which can occur to us. So before going further into the immune response against viruses, I will just talk about the basics of what are viruses and what are the different classifications of these viruses. Here you can see a picture, and from this picture you can understand like how much small a virus is. Here it is a human cell. It is 10 to 30 micrometer in size. Then this is bacteriophage, which is bacteriophage. I hope you know. It is a virus which is capable of killing the bacteria. Here the bacteria size is 2 to 3 micrometer, and here the bacteriophage size is much smaller, and it is 70 into 200 nanometer. In human viruses, you can understand it is smaller than the bacteriophage also. This is 20 to 30 nanometer picornavirus. It's much small. Then hypervirus is a little bit bigger, 162 to 30 nanometer. And HIV 100 to 130 nanometer. Then influenza virus 80 to 120 nanometer. So this much small compared to the human cell, which is 10 to 30 micrometer. So we cannot see the viruses in our naked eye. We have to use some uh, highly specialized microscope to see them. And viruses are very much small in size. These are the characterization of the viruses. They are small in size. They are obligate intracellular parasite, which means they are completely dependent upon the internal environment of the cell to create new infectious virus particles or virions. So they cannot survive in the environment They will only survive upon the host, and they will require the host system to create new infectious virus particles. So their genetic material can be composed of either DNA or RNA, but not both. So viruses are smaller in size. They constitute small segments of nucleic acid with protein or some lipoprotein coat, and they require the host system to replicate. So they can enter through the mucosa of the respiratory, urogenital, or gastrointestinal tract, and they account for most instances of viral transmission. Like through tract only, mostly viruses enter. They can also enter through broken skin, such as during insect bite or any kind of puncture wound. They normally can enter the host cell via one or more specific cell surface receptor for which they have affinity. They have some antigens on their surface which binds with the different kinds of receptors on the host cell surface, and through that they can enter into the cell. For example, influenza virus binds to some scalic acid residue in cell membrane glycoprotein and glycolipids. Rhinovirus binds to intercellular adhesion molecules or ICAMs, and Epstein-Barr virus binds to type 2 complement receptors on B cells. So you can understand the virus enter through the different kinds of receptors which are already present in our host cell. It is utilizing the host system only to enter, and through this attachment with the receptors present on the surface of our host cell, this virus can bind to and enter into our host cell. So it enters into the cell, it diverts the cell biosynthetic machinery to replicate itself. For example, the influenza virus genome replication step is often error prone, generating more mutations, but not in case of other viruses, because large numbers of new influenza viral particles are produced in a replication cycle. Many different mutants, some with survival and immune evasion strategy and advantages, can arise. So once it is entering through the receptor mediated endocytosis or any other mechanism through which it is entering into the cell, uh, it can it wants to replicate. No, the virus wants to replicate inside the host. So once it is entering into the host cell, it diverts the biosynthetic machinery to replicate itself. So the virus diverts cell biosynthetic machinery to replicate itself. So the influenza virus genome replication step, for example, it is taking the help of these viruses, will take the help of the cellular machinery like the cellular polymerases to replicate its own genome, because sometimes what happens, these viruses lack those systems, so they want to divert the host system so that they can replicate themselves. So in case of influenza virus, uh, it happens like the replication step is very much error prone, and there is no proofreading activity of the polymerase. So there will be too much mutation which is occurring, and whenever new virus particles are being made, there are too many mutations which are occurring inside the system, and like the viral particles will have too much mutation compared to the parent one, and they will produce new and new kind of strains. And many of those new strains can have more and more evasion and survival strategies, and they thus leads to different kinds of pandemics or epidemics, which is not good. So you can understand how the virus can harm our society. by generating new and new forms of mutants. Like in case of COVID also you could have seen, like new and new variants were arising every day, and those more and more what to say, bad variants, more and more harmful variants were arising. So there are different structures of viruses. For example, not all the viruses look similar. They have different shape, different size, even different genome, different like replication properties. So we need to understand what are the different kinds of uh, viruses and what is the mostly the common structure of the viruses. So there are three main things which are present in the viruses. For example, the we all know that the viruses, if it carries the naked like it is having a genome, but the genome needs to be covered. If we put a DNA or a RNA in the normal temperature, you think it will survive for a long time? No, it will degrade, or it will undergo some enzymes can come which can like uh, degrade it, or can chew the DNA or the RNA. So what it does, it is very clever. The viruses are, in order they want to protect their nuclear material from this harsh environment. So what they do, they surround the nucleic acid with a protein shell, which is known as the capsid. From it comes from the Latin word capsa, meaning box. So here you can see this is the genome, be it DNA or RNA. Can be double stranded or it can be single stranded. So what it does, it wants to protect its nucleus, its genome from the environment. So it forms a shell outside it. The shell is known as capsid. So this capsid is composed of one or more repeating protein units that create the entire capsid. For example, these one small round things are the capsid proteins, and these are in repeating nature, and they form the whole capsid, and they protect the uh, this genome. So it is very difficult to break this capsid open, and it protects the nucleic acid inside it. So the nucleic acid and the capsid together is known as the nucleocapsid. So this is the nucleus, sorry, not the nucleus, the nucleic acid. This is the capsid, and together they are known as nucleocapsid. Apart from this, there are some viruses which are enveloped. For example, there are influenza viruses which are enveloped, and they have an envelope surrounding the capsid. So it is also to for protection only, to give double protection, and mostly this envelope is created uh, by the membrane derived from the cell membrane, and most often it is plasma membrane, although the envelope can also come from the cell's endoplasmic reticulum, Golgi complex, or even nuclear membrane. Here you can see two types of viruses. One is naked virus. Here there are these capsid proteins. There are this nucleic acid genome, and it is protecting this capsid, and also there are virus attachment proteins to which it attaches to the receptors on the cell surface, and there this is this enveloped virus which is again surrounding the capsid. And here this this envelope generally is derived from the membrane of the uh, plasma membrane of the host cell, or it can be from derived from other membrane like endoplasmic reticulum, Golgi complex, or even the nuclear membrane. Here also you can see there are certain proteins, matrix protein, and this is also virus attachment proteins through which it can attach to the cell surface. So there are mainly three components. One is the nucleic acid, one is the capsid, and if envelope is present, then there is a envelope on the which is surrounding the capsid. So this is just an example of the shape of the different kinds of viruses. For example, there are helical viruses, there are rod-shaped viruses, there are circular viruses. Here you can see different helical viruses. For example, vesicular stomatitis virus forms bullet-shaped helical nucleocapsid. Here A. Then there is tobacco mosaic virus, which forms long helical tubes. Then there is Ebola virus, which is forming long threads that can extend over 1000 nanometer in length. And this is the icosahedral terminology and axis of symmetry. This is the icosahedron faces like fuchsia triangles, edges, red rectangles, and vertices, violet pentagons. And that is the whole head of the virus, you can tell. And there are different kinds of symmetry which are present. Like two, they can be two-fold, they can be three-fold, they can be five-fold, and example includes like two-fold in axis of symmetry means when the axis is placed through the center. When A is threefold means it is axis is placed in the center mm, of a face. Here you can see threefold like a triangle. Fivefold passes through the vertices of the icosahedron. It is like a pentamer kind of. So different kinds of axes of symmetry occurs. Different kinds of terminologies are there like pentagons, rectangles, triangles, etc. And these are different viruses are of different shapes. Then there is a classification known as the Baltimore classification. I won't go into details into the classification, but there are different kinds of viruses like the double-stranded DNA viruses whose genome is double-stranded DNA, single-stranded DNA are viruses whose genome is single-stranded DNA, double-stranded RNA viruses genome is RNA, double-stranded RNA. There are positive strand single-stranded RNA. There are negative strand single-stranded RNA. That means positive strand means directly it can translate into protein since from plus mRNA we can directly get protein. Negative sense means it first has to make the what to say its positive strand then has to get translated into protein. Then there are viruses that reverse transcribe. There are DNA viruses that reverse transcribe. So reverse transcription means RNA viruses they will form a cDNA and then they will replicate. For example, HIV is one of the class 6 virus. So different kinds of viruses are there which are according to their genome type they are divided into different classes. So these are the taxon, the examples. For example, there are order, family, genus, and species. Similarly, like what we have learned about taxonomy. So in case of order, it ends with virilis suffix. Only about half of viruses are currently classified in orders. For example, Picorna virilis is an example. Then comes family, which ends in VDD suffix. Subfamilies are indicated with VD suffix. For example, Picorna VDD. Like genus, which also ends in virus suffix. For example, enterovirus. Species means the common name. Example includes rhinovirus A and for example, you can see Picorna vidilis is the order, Picorna vigidi is the family, Picorna virus is the it will be the species, the genus, and obviously Picorna virus can also be the species also depending on the type. So this is the different kinds of taxa, different kinds of naming system like what is there, like family, class, order, family, genus, species in case of the normal taxonomic system. So here you can see they are categorized based on their type of nucleic acid. DNA viruses are there in this yellow box and RNA viruses in the blue box, and further they are characterized upon based on distinguishing characteristics. Virus taxa infecting vertebrates, for example, double-stranded DNA include adenoviridae, asperviridae, herpes virilis, herpes viridae, then papilloma viridae, iridoviridae, pox viridae, poliomyelitis. So all these viruses are DNA viruses. These are having double-stranded DNA in their genome, so they belong to class 1. Then single-stranded into Aniloviridae, Sarcoviridae, Parvoviridae, then Hepadnaviridae. So is a double-stranded DNA virus, class 1, and it can get reverse transcribed. So it will go to the class 7. And here you can see this single-stranded will go to class 2. Then comes the RNA viruses, can have negative strand or positive or negative strand. For example, uh, adenoviridi, buniaviridi, delta virus, orthomixoviridi. These are having negative strand, and these are mononicoviridalis, bornaviridi, paramyxoviridi, rhabdoviridi. They can have a positive or negative strand. Then only positive strand single strand, that means they belong to class 5, is class 5. Then class 4 includes positive strand, which includes coronaviridae, arteriviridae, nidovirilis, astroviridae, calciviridae, picodum, picomaviridae, togaviridae, etc., flaviviridae. So they are in the class 4. Then there are double-stranded RNA viruses like Brinaviridae, Picomaviridae, Spinoidoviridae, Cedaroviridae, which is class 3. Then remains ssRNA, or which can reverse transcribe. Example is Metaviridae and Retroviridae, as I told, HIV, which belongs to class 6. So these are the different types of viruses belonging to different classes, different types, having different genomes, and you can see all are looking different from one another. Not one will look similar to one another. Maybe even if they look similar, they are different. Is there in size. So after this, uh, first I will discuss about the doubt which someone was having before going on to the innate immune response to the viruses. Okay, so doubt was regarding susceptibility to antigen processing question. Why will the L amino acids induce immunogenicity and not the D amino acids? We learned about foreignness, that is lesson related to the antigen. More will be the immune response. Here D amino acid should be inducing more response, but they are not. But L amino acids are. Why? Because we know that in nature most of the proteins have this L amino acids, and this D amino acids presence in protein is very much rare. So uh, these L amino acids can induce in immunogenicity, but why not D amino acids? So these D amino L and D are based on the chirality of the molecule. So it has been seen that these D amino acids are resistant to the proteases which are present in our body or the system. So this, if the proteases cannot act on that D amino acid, it cannot be processed further. So what happens in case of L amino acids, the proteases can recognize, and it can target it and can chop it into small pieces, and it can be presented via the MHC class 2 molecules to the T helper cells, and that can induce the immune reaction. But in case of D amino acids, due to the chirality of the nature, most of the Proteases cannot recognize it and cannot process it. It cannot be chopped into or break into smaller antigens, like it cannot be processed by the proteases, and it cannot be presented to the class 2 molecules by the MHC. Sorry, it cannot be processed and it cannot be presented to the TH cells via MHC class 2 molecules, and so it cannot induce immunogenicity, but there are certain like cases where it has been seen to induce immunogenicity, but not through this classical MHC class 2 pathway. But it compared to the L amino acid, the immunogenicity induced by this D amino acid is much lesser, and so it cannot induce more immune response. And while L amino acid can induce more immune response. So first, innate immunity to virus infection. So we all know that innate immune immunity is the first line of defense. So this innate immunity can itself first clear the infection without the help of any adaptive immune response. So first we are going to see uh, what is the innate immune response to virus infection. I have already taught, Sir has also taught about PAMs and PRRs. There are pathogen associated molecular patterns which are present on the surface of the uh, pathogens, which are recognized, like, for example, LPS or any kind of RNA, double stranded single stranded RNA, which are recognized by the PRRs, or the pattern recognition receptors, which can recognize these patterns and they can induce the immune response. So, for example, one of the PRRs are the Rigai molecule, which recognizes 5-triphosphate RNA and short DS RNA, whereas MDA5 discriminates long DS RNA generated during the course of virus infection. Here you can see this is a Rigai. You can see you already know the domains, like the card domain, which is known for oligomerization. One person has a question who also gave a doubt last week, I think last to last week, like what card domain is used for. It is not used for signaling, it is used for oligomerization. Then after oligomerization only it can induce the signaling. So this you can see this Rigai can sense this triphosphate, 5' triphosphate RNA and short double-stranded RNA, where another PRR, which is MDA5, can recognize the long double-stranded RNA. Here you can see the examples. Long double-stranded RNA are produced by picornaviridae, EMCV, Thieler's virus, Mengo virus, while short double-stranded RNA or triphosphate RNA can be produced by paramyxoviridae like Sendai virus, Newcastle disease virus, orthomyxoviridae like influenza virus, rhabdoviridae like vesicular stomatitis virus, flaviviridae like Japanese encephalitis virus, hepatitis C virus, and this 5' triphosphate RNA can be produced by flaviviridae like West Nile virus, dengue virus, Rioviridae like Rio virus. Here you can see MDA5 and Rigai, which is sensing these two types of different types of RNA. Uh, so they are differently recognized by Rigai and MDA5. For example, Rigai is responsible for detecting paramyxoviridae, orthomyxoviridae, rhabdoviridae, and some flaviviridae members. Not both cannot recognize all the viruses. Some recognize some viruses, and another recognize other viruses. So Rigai is responsible for paramyxoviridae, orthomyxoviridae, rhabdoviridae, and some flaviviridae, while MDF5 can recognize this picornaviridi, and some viruses such as West Nile virus and reovirus are detected both by RIGI as well as MDA5. And LGP2 is reported to be a genetic regulator for the RIGI and MDA5 signaling, like LGP2 molecule can act as a regulator for both of these signaling. So here you can see both this RIGI as well as MDA5 are recognizing different types of viruses, and they are initiating different kinds of response, which I will tell. So there are three classes of PRRs for RNA virus recognition. There are toll-like receptor, there are RIGI-like receptor, there are node-like receptors. Sir also has already taught about all these receptors previously, and I have also added some slides in the previous classes regarding all these receptors. And there are this, for example, the here you can see TLR pathway, here RLR pathway, NLR pathway. Like for example, a single-stranded RNA from viruses is recognized by TLR7 in PDCs. Here you can see single-stranded RNA from viruses. This is TLR7, which is PDC means plasmacytoid dendritic cell, which is a type of dendritic cell. This is recognized by TLR7. Then double-stranded RNA is detected by TLR3 in conventional disease. Then TLR7 and TLR3 signaling cascades include adapter, mediative as well as strip. So here it will be, I think, single stranded, not double stranded. So by mistake it has been labeled as double stranded. Double stranded is recognized by TLR7. Sorry, single stranded, and double stranded here is recognized by TLR3. And this mediated is adapter for this TLR7, while this strip is adapter for TLR3. And here it is activating the NF-kappa B pathway, which is inducing the cytokine genes. So releasing this I kappa A and beta, these are the negative regulator of NF-kappa B. They are releasing the NF-kappa B molecule, which is going to the nucleus and transcribing the cytokine genes. For example, then comes the RLR pathway, or the RIGI-like receptor pathway, which, as I already told, RIGI can sense this triphosphate RNA as well as the short double-stranded RNA, while long double-stranded RNA are recognized by MDA5. They both act and recruit one adapter molecule, which is IPS1. This IPS1 can also strip as well as IPS1 share signaling molecules for phosphorylation of IRF3 and IRF7, like TBK1, I-kappa, and mediated dependent signaling, which directly activates IRF7 in the PDC. So here you can see IPS1 can directly act on this I-kappa molecule and activate the NF-kappa B signaling pathway. TRIP can also activate the TBK1 pathway, and there can be activation of IRF3 and IRF7, which can induce the expression of this type 1 interferon gene. So all these signalings are interrelated. IPS1 can activate this pathway, TRIP can activate this pathway, mediated also can activate this pathway. So phosphorylated IRF3 and 7 can activate the expression of type 1 interferon genes. TLR and RLR induce the translocation of NF-kappa B to induce expression of cytokine genes via distinct adapter proteins. So one of the NLR proteins is there in case of NLR, or node-like receptor, in which one's name is NALP3, detects the presence of double-stranded RNA and induces the activity of caspase-1 via an adapter which is known as ASC. Caspase-1 is essential for cleavage of pro-IL-1 beta to IL-1 beta. Thus, it is also a cytokine. So, you can understand this is completely different from the TLR and RNR pathway. It is activating the caspase pathway, which is activating the IL-1 beta. So, this is a different pathway. This is just a short representation of all the TLR MLR pathway. But there are other molecules in all the three pathways which can activate other pathways as well. So this is the innate immune response for virus recognition. Apart from this, also many molecules are there which can actually help in the recognition of this virus and activate different signaling pathway. But I will talk about it in the next class. Apart from that, there is also adaptive immune response which can respond to viruses. For example, there is humoral as well as cell-mediated immune response, which has different responses against viruses. Humoral means, as you know, body fluid, and it includes obviously antibodies. So there are antibodies like IgA, which blocks binding of virus to host cell, preventing infection or reinfection. Then there is IgG, IgM, and IgA antibody, which blocks fusion of viral infection envelope with host cell's plasma membrane. So if it is blocking fusion, it will block the entry. Then there is IgG, IgM antibody enhance phagocytosis of viral opsonization, which I've already taught in the complement class. There is IgM antibody which agglutinates viral particles. IgM forms a pentamer and it agglutinates the viral particles. There is complement activated by IgG, but the antibody pathway where it is complements are activated, and it also mediates opsonization of the virus by superior lysis of the environal viral particles by forming that MAC or the membrane attack complex. So these are the different functions played by the different antibodies, and how they remove the virus from our system. Then there is cell-mediated response like the IFN gamma is secreted by TH or TC cells, and they can directly kill the antiviral active. They can directly kill the virus. They can have different antiviral activity. There is cytotoxic T lymphocyte, which is receiving the signal from MHC class 1 molecule via the APCs, and they can kill directly the virus infected cells. Then there are NK cells, natural killer cells, and macrophages that they kill the virus infected cells by antibody dependent cell mediated cytotoxicity. This is a phenomena which we can learn later. And as you can see there, these are in short the different kinds of immune response to viruses by the humoral as well as the cell mediated, means the adaptive immune response. So uh, that is all for today. And if anyone is having any question, they can ask directly in the portal, or like last week, I think someone has asked question. I will discuss it here. I will also give reply in the main portal, and also if anyone has any question, you can mail me, or you can directly ask in the video section of sir. So, or in my comments also you can ask. So I hope anyone, if anyone has any problem with the assignment, especially, please don't feel shy to ask anything, because assignments are important, and you can ask it directly through the class also, through the portal also, or through the comments also. So thank you for listening today. Uh, Thank you.